Students, in the last part we have studied how Cohen and Boyer they have developed their first artificial recombinant DNA. Actually, what's the process of making recombinant DNA? Here you can understand the word recombinant DNA. It is rDNA. Let us see the process of biotechnology. There are three steps of the process of biotechnology. The first step is, consider this is the plasmid. Single circular. It's a extra chromosomal part, so it is present in the bacteria. So it's a circular DNA. This is our gene of interest. This part you can say the gene of interest that we need, our gene of interest. So first isolate the gene of interest. So we have to identify where our gene of interest is there, then we have to isolate it. We have to extract it. So there are some tools are there in biotechnology. We have to cut it. So we have to isolate. This is the another extra chromosomal, circular chromosomal part. Here the same part we have to cut out. This part we have to cut out, and this our gene of interest we have to join here. By the help of molecular glue, we have to join here. So that the second step we can understand, insert the gene of interest into the plasmid. So this is our plasmid, our gene of interest we have inserted. Now this is called rDNA, or you can say recombinant DNA. Now this recombinant DNA we have to insert into the host. So the third part is insert the plasmid into the host. So these are the three steps of the process of biotechnology. Here you can understand there are some tools are necessary for the biotechnology. So in this session, in the part we are going to study about what are the tools of biotechnology. So we can understand what are the tools of biotechnology. We are using, there are some enzymes like restriction enzyme, and the another one is ligase and vector, and another one is host. So these are the tools of biotechnology or recombinant DNA technology. So we are going to study one by one. First of all, we can understand what is the meaning of this restriction enzyme. So, what is restriction enzyme? By the help of the word you can understand. Restriction, something it is used to restrict. Yeah, so restriction enzyme actually it is present in the bacteria. It is present in bacteria, and it is used as protection, or it is used as defense against the bacteriophage. It is used to kill the bacteriophage. It is used to lysis the bacteriophage. So it is present in the bacteria only. At first, in the year of 1963, one of the scientists, his name is Arber. He introduced the first restriction enzyme. He isolated the restriction enzyme, two different types of restriction enzyme. It is present in E. coli. It is used to restrict the growth of bacteriophage. So, what are those two restriction enzymes? One, he has added to the methyl group. One, it is added to the methyl group, and another one is it is used to cut the DNA, that is called restriction endonuclease. So this is the discovery of restriction endonuclease. Next we can understand the first restriction endonuclease is HIN2. HIN2 is the first restriction endonuclease. See, once they have extracted this HIN2, then later after 5 years they have have identified where it can be used, which part of the DNA can be cut. So that particular site, particular restriction enzyme, it can be cut in the particular place only. So that site you can understand recognition site. It is called recognition site. So particular place only it can cut. So it can understand, it can recognize and it can cut. So that you can understand recognition site. So before we are studying about all these recognition sites and restriction enzyme, we can understand some more detail about the restriction enzyme. Restriction enzyme, it is included under the class of one of the class. It is called nuclease. Under the class of nucleases, it's one of the important question for the board as well as NEET. Under the class of nucleases, there are two different types of nucleases: endonucleases and the another one is called exonucleases. So endonucleases and exonucleases. So this remaining we can study in the next part. Thank you, students.